Welcome back to the deep dive, everybody. Today we're going deep on how organisms create those compounds we've all heard about. Secondary metabolites.
Yes, these compounds are incredibly important.
They give us vital medicines. They're behind some really potent flavorings. There's just a lot going on there.
Yeah, and that's what we are going to unlock today, the secrets of how organisms make them.
Yeah. And so our mission is to really try and understand, like what are the genetic mechanisms? Like how is this actually happening? And, how are scientists sort of getting in there and manipulating these processes?
Yeah, we're going to look at the blueprint,
Okay.
the genetic instructions, if you will.
All right. So when we talk about that blueprint, like how important is an organism's genes to determining, can they even make the compound? And then like how much of it can they make?
Whether everything. The organism's genetic code. It's the foundation for whether or not it can even make a secondary metabolite and how much. You can almost think of it as like a recipe book, right? Where pathway genes are the specific instructions for each step in building that compound.
Okay.
And much like a recipe, right? If you have slight variations in these genes, maybe you substitute almond flour for regular flour, these are called alleles.
Okay.
And that's going to give you a slightly different product
Uh-huh.
or maybe a different quantity.
Okay, so that makes sense. So just thinking about it, you've got, millions and billions of base pairs in DNA. It's not like these pathway genes are just like randomly scattered throughout. How are they organized?
That's right. They're not just randomly floating around.
Oh,
In many organisms, especially microbes, these genes are grouped together
okay.
in what we call gene clusters.
Okay.
And this is really efficient because it allows coordinated control. You can turn the entire pathway on or off as needed.
Yeah.
And it also, because of this close proximity, it actually can facilitate sharing of genetic information between organisms.
So I'm thinking about it like, okay, so we've got this organization, and you can kind of see how scientists can start to understand, like, okay, this is how it's organized, this is what we can target. And I think that leads us to this whole idea of metabolic engineering.
Right.
Yeah.
And so once we identify those genes, we can make very targeted modifications.
Okay.
That's what metabolic engineering is all about.
Okay.
You find, for example, maybe you have an enzyme in there that's really crucial, but it's kind of slow.
Mm-hmm.
It's slowing the whole process down. So what we can do is increase the number of copies of its gene.
Oh, okay.
And that would lead to, essentially boosting the production.
Sure.
And this has been done. You see huge increases in the yield of really important drugs.
Like adding more workers to a bottleneck.
Exactly.
Okay, cool. So what are some other types of tweaks that we're seeing?
So we can also introduce entirely new genes, even whole pathways into an organism, right?
Yeah.
That gives it the ability to create novel compounds.
Mm-hmm.
Or if you've got, let's say, competing pathways in there, all vying for the same resources,
Yeah.
you can disable their genes.
Okay.
And so that way more resources are funneled towards the metabolite you want. And then you've also got the manipulation of regulatory genes, right?
Okay.
These are the ones that control the activity of all the other genes in the pathway. You can tweak those to increase the output.
So it really is like having control of a factory at like the micro level.
Absolutely.
So I think one of the things that I really thought was fascinating, and you mentioned regulatory genes, was this whole idea of transcription factors, and the source material really highlighted these as kind of the puppet masters.
Yeah, they're really interesting.
Yeah.
So transcription factors, they are proteins that can bind to specific DNA regions, and they're kind of like a switch, right?
Oh, okay.
They control how much of a gene is expressed,
Okay.
how much of it is actually made into a functional protein. And what's really key is that a single transcription factor can actually regulate multiple genes
Oh, wow.
within an entire pathway.
So they're coordinating the whole show
Exactly.
for making a specific compound.
Yes, and they respond to signals, right?
Okay.
Like what stage of development is the organism at? What's the environment like? Is there light? Is there stress? Are there specific molecules present?
Okay, so they are really kind of in there fine-tuning everything.
Yes, absolutely. And in plants, for example, families of transcription factors like MYB, these are key regulators of flavonoid production.
Wow.
And flavonoids, they're responsible for those vibrant colors in flowers and fruits. They have antioxidant properties.
Okay.
So they're super important.
Yeah.
And so manipulating these transcription factors is a really powerful way to just like crank up that output for a whole pathway.
Okay, so let's kind of take a step back. We've talked about, genes, we've talked about how they're organized, and then we've talked about how scientists are manipulating them. So the genetic factors are really the instructions, right, for creating all these secondary metabolites.
That's right. And they work with environmental and developmental cues.
Sure.
But the genes, they're the core capabilities, right? And they're the control mechanisms.
And this is being applied in the real world.
Absolutely. Things like selective breeding, inducing mutations, and modern genetic engineering, all of these leverage these principles to improve yield.
Okay. So for all of you listening out there, the key takeaway here is that an organism's genes are a primary driver for the production of these incredibly useful compounds. And there are huge opportunities here for optimization.
Right. It's a really exciting field.
It is, and I'm kind of left wondering, considering how precisely we can now manipulate these pathways, are there some completely unexpected areas where this knowledge might lead to breakthroughs?
I think there's a lot of potential there.
All right, thanks for joining us, and we'll see y'all next time.
Thanks for having me.